Welcome students. Today I will be teaching you fundamental unit of life, cell, of class 9th. Let us start studying about how cells were discovered. Many scientists were continuously working to improve the microscopes because the cell is so small that it cannot be seen with the naked eye. There are examples of cells which we can see with naked eyes, but mostly all the cells are very small and they are microscopic in nature. The scientists were working hard to improve the microscopes. There were various scientists who had contributed towards the cell. They are Robert Hooke, Leeuwenhoek, Schleiden, Schwann, Virchow. The first cell was discovered by Robert Hooke in 1665. He said that under the microscope, the cells looked like small rooms in which the monks used to stay. The cells that he saw were the dead cells, and they were taken from the plant cells, that is the cork cells of the plant. The cork cells are found on the bark of the trees, the outer part, the dried part of the tree's trunk. You can see the cork cells over there. So Robert Hooke was the first person who discovered cell in 1665. Now let us see how different scientists contributed in biology for the study of cells. 1665, Robert Hooke saw the cells in the cork. In 1674, Leeuwenhoek discovered protozoa, and later on, he also saw bacteria some 9 years later. In 1833, Robert Brown described the cell nucleus in the cells of the orchid. In 1838, Schleiden and Schwann proposed cell theory. 1858, Rudolf Virchow, he gave a famous theory, omnicellular acellular, that is, cells developed from pre-existing cells only. Let us study about the modern cell theory. The cell theory says that all the living things are made up of the cells. The cell is a structural and functional unit of a living organism. The new cells comes from the pre-existing cells by the process of cell division. These cells, they carry hereditary information from one generation to another generation. The composition of all these cells remains the same, and all energy flows of the life occur within the cells. Let us study about the shape and size of the cells. All the cells do not have the same shape. The shape varies, the size varies. The shape and the size depends upon the function that they are performing and place where they are found. See the red blood cell? It is a spherical in nature. We say it as biconcave with little depressions in the middle. This is a smooth muscle cell. It is a spindle shape. Spindle shape is a shape of your eye. You can see on the side, the sides are very narrow. The middle level is thick and the ending is again very thin and narrow. This is a guard cell. This is a stomata, which you can see in between the cells of the guard. This is a nerve cell. It is the longest cell of our body. The paramecium is a unicellular organism and it looks like a slipper shape. And this is a diatom. a type of an algae. Now, you can see the smallest cell. It is called as PPLO. It is a type of bacteria only. It is prokaryotic in nature. The largest cell is an ostrich egg. The longest animal cell is a nerve cell. Okay, and there are some cells that can change the shape continuously. Amoeba. We also have WBCs, white blood cells. And these white blood cells are of different types. And in each of different types, the shape of the nucleus also varies. So the shape, the size, depends upon the specific functions that they are performing. Remember that these cells size remains the same. The RBC size will remain same in different animals. It is not going to change. Let us take an example of an elephant and let us take an example of us. The size will not vary. Yes, the number of cells will vary because the size of the organism is such huge. So the number of RBC will change, but the shape and size will not change. So here again, you can see that this is a stem cell. Stem cell is an undifferentiated cell, as it can differentiate into different types. Different types means undifferentiated means it can take the shape and size of different types of cells. So a stem cell can change into a muscle cell, a stem cell change into a sex cell, a stem can change in an immune cell, that is a WBC, a nervous cell, a blood cell, bone cell, or a fat cell. You might have learned about the many stem cells bank that is there. From the placenta, the human cord, the stem cells are preserved in the bank. Or size cell called as a structural and functional unit of any living organism. A cell is called as a structural part of any living organism because just as a brick in a building is a unit of that building, the cell is again a unit of our body. Therefore, it is called as a structural unit. Why a functional unit? Each cell, inside each cell, there are different small body parts that are called as organelles. And each of these organelles perform different functions of transportations, excretions, and various functions that our whole body's different organs are performing. So we have got an organ that is called as the circulatory. We have got a system that is called as a circulatory system in our body, and the whole of the blood and lymph flows in this circulatory system. In the same way, inside the cell, the endoplasmic reticulum does that part. So inside the cell, there are few small body parts, very small, that do their own functions. They perform all the functions that is necessary for the life. And therefore, it is also called as a functional unit of living organism. Organisms show variety in cell numbers, shape, and size. We have already studied regarding this, that the number of cells vary in organisms. It depends upon the size. The shape and size remains the same. Only the number will change, and it depends upon the size of the organisms. Can you guess the number of cells in a tall tree or in a huge animal like an elephant? 1 billion is 1,000 million, and 1 trillion is 1,000 billion. The number runs into billions and trillions. The human body has trillions of cells, which vary in shape and size. Different groups of cells perform a variety of functions. The size of the cell is limited by the ratio of volume to the outer surface area. The larger the cell, the larger the surface area that must be maintained. Cell shape is typically related to the cell function. Most of the cell shapes are roughly cuboidal or spherical. Some cells have long extensions to reach out in different directions. For example, the nerve cell, a neuron. The shapes of the cells can thus be called as spherical shape. The example is RBC, a spindle cell, that is a smooth muscle cell, an elongated cell, that is, they are shaped like a branch of a tree, for example, a nerve cell in humans. The cells are broadly divided into two types, that is the prokaryotes and the eukaryotes, and it depends on the types of nucleus that they are having. If the nucleus and other organelles, they don't have a membrane around it, then they are prokaryotic cells. So prokaryotic cells have membranes that, prokaryotic cells lack membrane-bound organelles. Example, bacteria is the only organism that have prokaryotic cells. Eukaryotic cells contain organelles that are bound with membranes. And all the cells other than the bacteria are eukaryotic cells. The nucleus has also a membrane around it. Since in prokaryotic cells, the nucleus do not have a membrane around it, it is also called as nucleoid. A prokaryotic cell does not have internal organelles surrounded by a membrane. The organelles that are found in the prokaryotic cells are the ribosomes, DNA, a plasma membrane, a cell wall. The eukaryotic cell have a distinct membrane-bound organelles, and they allow cells to perform different functions. The organelles are nucleus, nucleolus, chromosomes, plasma membranes, and various other parts like the endoplasmic reticulum, then your mitochondria, etc. This is how a eukaryotic cell is divided. It is mainly divided into three parts: a cell membrane, that is a membrane that is found on the outer margin of the cell. Then there is a nucleus in the center, which is bound with the nuclear membrane. And between the nucleus and the cell membrane is the cytoplasm. Now, what is protoplasm? Protoplasm means this is all the matter that is found in the cell. It includes everything that is a cytoplasm and a nucleoplasm. Nucleoplasm means it is again a base material that is found inside the nucleus. Like the cytoplasm and the nucleoplasm together form protoplasm. So a cell is broadly divided into three parts. The eukaryotic cell. Whenever we talk about cell, we say a eukaryotic cell. So it will have a cell membrane, which is also called as a plasma membrane, a cytoplasm, and nucleus. The protoplasm is broadly divided into two parts, the cytosol and the cell organelles. These cell organelles are broadly divided into two types: the organelles which do not have a membrane and the organelles which have a membrane. Remember, as I told you, that the prokaryotic organisms have got organelles which do not have membranes. But the eukaryotic cells have organelles which have membranes and which do not have membranes. They contain both the type of organelles. So let us see the list of cell organelles which do not have membrane, and they are found in a eukaryotic cell. Ribosome, it is found in prokaryotic and eukaryotic both together. Centrioles, they are found in eukaryotic. The microtubules, the microfilaments. The cell organelles which have got membrane are broadly further divided into two types. Only one membrane is found around the cell, that is called a single membrane, and a double membrane is also found around the cell. So the double membranes are mitochondria and chloroplast. Rest of all the organelles are single membranes. They are Golgi body, the endoplasmic reticulum. The endoplasmic reticulums are rough endoplasmic reticulum and the smooth endoplasmic reticulum, lysosomes, vacuoles, peroxisomes. See how the plasma membrane look like. It is a double membrane layer, and this blue color two membranes are made up of lipids. In between, it has got proteins. So you can see this is a protein, this is a protein, this is a protein, these are proteins. So in between the two bilipid layer are the protein layers found. These proteins are further divided into two types. They are extrinsic and they are intrinsic in nature. There are different types of proteins found here. One which is found only in the outer membrane, one which is found only in the inner membrane, and one protein that is found throughout from the outer membrane to the inner membrane. Remember, see the shape? They are pathways for regulation of entry and exit of materials inside and outside the nucleus. Sorry, inside and outside. They are the pathways to regulate the entry and exit of materials inside and outside the cell. So the structure of plasma membrane, it is made up of lipids, that is a fat, and proteins. This lipid is bilayer, and in it are proteins embedded in it. Transport in cells across the plasma membrane. The transportation in the plasma membrane is done by diffusion, osmosis, and active transport. Diffusion is the movement of particles from a high to lower concentration. And osmosis is the diffusion of water across a membrane. Active transport moves particles from lower to higher concentration. You should clearly know the difference between all the three. So diffusion, what happens? The particles are moving. For example, let me take a spray that is in your bottle. So in the bottle, the concentration of the spray is very high. And when you are spreading the scent, what happens? It is moving out. So the particles are moving out from higher concentration to the lower concentration. Diffusion takes place both in living things and non-living things. I gave you the example of non-living. Let me give you an example of living thing. How do we take in oxygen and how we give out carbon dioxide? It is because of the process of diffusion. Same way, even plants take in oxygen and give out carbon dioxide, or even for photosynthesis, plants require carbon dioxide and they give out oxygen by the process of diffusion. In osmosis, the particles are not moving. The water will move across a semi-permeable membrane. That is the place from where the water is more to the place where the water is less. And in active transport, the particles are going to move from a region of low to higher concentration. See diffusions, the particles are moving from higher concentration towards a lower concentration. And the movement is random. And it is a passive process. Passive means no energy is required for the particles to move. See, a drop of ink when it is put in water. Here the concentration of ink is more. As we are putting in water, it is spreading, it is diffusing. And see how it is diffused from a region of higher concentration to region of lower concentration. See over here, again you can see this is higher concentration and now it is diffused in the whole of this beaker. Osmosis is the movement of water across a semi-permeable membrane from a region of high water potential to a region of low water potential. So from here, you can easily see that here the water content is more and here the water content is less, and in between is a semi-permeable membrane. What happens after some time? The water will move across to here. See, the water has moved across here. The water level is increased. Okay, till a balance is done. Here also you can see this is a semi-permeable membrane. These are sucrose molecules and these are water molecules. Where the water molecules is more, the water molecule is more over here. You can see the number 1, 2, 3, 4, 5, 6, 7. Here there are more water molecules. Naturally, water will move from this side to this across a semi-permeable membrane, and after some time the water level on this side will increase. Again, the examples of osmosis. In osmosis, the solutions are broadly divided into 3 types of solutions. They are hypertonic solution, they are hypotonic solution, and they are isotonic solution. Let us start with the hypotonic solution. If RBC is put in a hypotonic solution, that means simple water, what will happen? After some time, the water will enter inside the cell and it will swell. We do not have a cell wall. So what will happen after some time? It can burst. Second, the isotonic solution. That is the water content inside the RBC and outside the RBC cell is equal. And therefore, how much amount of water will go inside, that much amount of water is going to come out. And in hypertonic solution, you can take a salt or a sugar solution outside and put a cell wall, blood cell in it. After some time, the water will move out. And what will happen? The red blood cell will shrink. So this is a hypertonic solution. So this is you can see the cell has shrunk, the solution is hypertonic solution, the water has moved out. Isotonic solution, the cell has remained as it is because how much amount of water is going inside, that much amount of water is moving out because the water level is balanced. And over here in hypotonic solution, the water is moving inside and therefore the RBC has bulged. So the 3 types of solutions are hypertonic solution, hypotonic solution, and isotonic solution. Hyper means the concentration outside the cell is more. Hypo means the concentration inside the cell is more. And isotonic, the concentration of water inside and outside is going to remain the same. What happens when osmosis takes in a plant cell? Plant cells have got a cell wall that protects the plasma membrane. In isotonic solution, the water inside and going outside remains the same. In hypotonic solution, water enters inside the cell. But after a certain time, the pressure will be given by the cell wall to the plasma membrane, and the plasma membrane will give the pressure to the cell wall. So it won't burst like that. It is taking place in the human cell. And hypotonic solution, the water is moved out. The plasma membrane has moved inward away from the cell wall. we call this as plasmolysis. So there are certain terms that you should be knowing in animal cell and plant cell about what happens when they are in hypotonic solution, hypertonic solution, and the isotonic solution. When water enters into the animal cell and it starts bulging, it is called as lysed in animal cell, and then it is called as turgid in plant cell. So you can note the meaning of turgidity. Turgidity means the water is entering inside the cell. When the solution is isotonic solution, it is called normal in animal cell and flaccid in plant cell. And when the solution is hypertonic solution, the animal cell loses of water and it is called as shriveled, and the plant cell is called as plasmolyzed. Under this condition, the plant cell is going to die, and even during the lysed condition, since it is bursting, the animal cell will die. Let us study what is plasmolysis. Plasmolysis is a shrinking of cytoplasm away from the cell wall. See, this is a cell wall and the plasma membrane was very close to it. Now the plasma membrane is shrinked and it has moved away from the cell wall, and this is called as plasmolysis. It occurs when the plants are placed in dry region or in a very high salt environment. It causes cells to lose turgor pressure and causes the plant to wilt. Wilt means the plant is drooping down and the leaves will die. The cell walls that are found in the plants. So you can see the cell wall. This is not found in animal cells. The cell wall which is found in the plant cell is a permeable membrane. Permeable membrane, water can enter through it. These parts, even between how there can be holes in between, pores we call this, called as. They'll allow the things or substances to enter inside to the plasma membrane and inside the cell, and also exit. Functions of the cell walls. They prevent enlargement of the plant cell. They play a very important role in absorption, transport, and secretion of plants. They provide the plant cell a definite shape and size so that they get strength and rigidity. They protect the plant from physical injury, and together with vacuoles, they provide skeletal support. Osmosis in potatoes. Now, let us take a potato plant and let us peel the skin from outside and make a small hole in it. Now, this is the original potato cylinder that we have made. Now, let us take different beakers. In beaker 1, the potato is kept in a pure water. In beaker, let us study about osmosis in potato. Let us take 2 beakers, that is a beaker B and beaker D that we have taken over here. Peel a potato like from outside and make a shape like this. And even you can make a small hole in between. Now put this potato in pure water and put this potato in a dilute sugar solution or dilute salt solution. After some time, you will see that the potato that was in beaker B has swollen. You can see how it says in C. This has swollen because it has absorbed water. The water is entered inside the cell. What happens to the potato that was kept over here in the sugar solution? It is shrinked because it has lost water. This is an example of osmosis in potato. What will happen if I take a boiled potato? Osmosis will not take place So osmosis takes place only in living things. The boiled potato is dead. The simple potato is still living. You can see the birds growing during the moist season, that is the rainy season, the moist season. That is, it is growing, it is living. So the cells are living. So osmosis is seen only in living things. Diffusion can be seen both in living things and non-living things. Let us take another example of egg. I can remove the eggshell. You have got a vinegar or acetic acid at home. Put the egg in that acid and you will see after some time the whole of the calcium carbonate layer, that is the hard shell, is removed. And you can see a cell like this. Carefully pick it up and put it in two types of water. One in a normal water, distilled water, and one in a salty water or a sugar water. What will happen? See, this is in sugar water or a salty water. It is shrinked. And this has bulged. And this has remained. If you are kept in a normal water, in a isotonic solution, it will remain as it is. So this is kept in a, egg is kept in a hypertonic solution. This is kept in a hypotonic solution. And this is kept in an isotonic solution. So osmosis in boiled potato will not take place as the cells are dead. Diffusion can take place both in living things and non-living things, but osmosis will take place always in living things. So if you set up a boiled potato in a hypotonic solution, in a hypertonic solution, and in a isotonic solution, no change will take place because the cells are dead. So again, diffusion versus osmosis. I've highlighted this again and again, the difference between the diffusion and osmosis, because you should know, because this topic will again and again come in science, in chemistry, in physics, in bio. It will come in different topics. So let us see what happens in diffusion. The solutes, that particles are moving from a region of high concentration to lower concentration. It does not require any energy at all. Osmosis, what is happening? The solvents are moving from a region of solvent. See, solute, solvent. Solute means this particle, and solvent means the water. Water is moving from a region of higher concentration to lower concentration across a semi-permeable membrane. The water content has risen on this side because it has moved towards a low concentration side until an equilibrium is obtained. Equilibrium in water, equilibrium in solvent. Here equilibrium, the whole of the solution, equilibrium in the solid particles, in the whole of the medium. So the whole chart which shows the differences between diffusion, osmosis, and active transport. Diffusion takes place by movement of molecules, osmosis by movement of water molecules, and active transport by particles of a substance. Diffusion takes place from a region, molecules move from a region of high concentration to lower concentration, and osmosis, the water molecules move from a region of higher molecule concentration to a lower water molecule concentration, and active transport takes place from a region of lower concentration to higher concentration. Active transport means energy is required over here. The structure and function of the nucleus. Now we will study the plasma membrane and the process of diffusion and osmosis taking place across the plasma membrane, and how entry and exit of materials takes place by the process of diffusion and osmosis across the plasma membrane. Now we will study about the next topic, that is the nucleus. So inside the cell, we find a nucleus, and this is a nucleus. We will be studying what are things are seen inside the nucleus. Robert Brown discovered nucleus. A definite nucleus is absent in prokaryotic, means the nucleus without a nuclear membrane is found, not with a nuclear membrane. Instead, a DNA is found in a prokaryotic cell. If a definite nucleus, that is a nucleus with a nuclear membrane, is found in the cell, it is called as a definite nucleus. And they are found in the eukaryotic cell. The position of the nucleus in the cell is definite. It occupies more or less the central position. Now and then it is pushed to die in the peripheral region. Two types of cells are there, prokaryotes and the eukaryotes. Prokaryotes, they are one-celled organisms with no membrane around the nucleus or any other organelles. And eukaryotes are multi-celled organisms with membrane-bound nucleus and other organelles. So this is a eukaryote. You can see eukaryote nucleus. Only I've showed you the nucleus part, not the whole of the cell. So now you have to concentrate only on the nucleus. See, the nucleus, they have got a nuclear pores and they have got chromatin. This is chromatin. The nuclear pores allow the entry and exit of materials inside the nucleus and outside the nucleus in a cell. Chromatins are nothing but they are chromosomes. When the cell is not dividing, the chromosomes are found in this network structure, and that is called as chromatin. Nucleoplasm, nucleoplasm, it is the base material that is found in the nucleus, and nucleolus, it helps in formation of ribosomes, and also it synthesizes RNA. So here you can see again that the same nucleus that I've talked about. This is a nuclear pore, allows entry and exit of materials. This is a chromatin. This is a nucleolus. Nucleolus helps in formation of an organelle that is called as ribosomes, and also synthesis of RNA. And this is chromatin. Chromatin is found when the cell is not dividing. So it is made up of DNA. So a nucleus is not that only one nucleus is found in an organism. Sometimes a nucleus is absent in certain cells. In us, in human beings, in our RBC, no nucleus is found. Sometimes, or most of the time, only a single nucleus is found. Sometimes two nucleus is found, for example, of cartilages. And sometimes many nucleus are found, for example, in our osteoblast. It is called as polynucleate. Poly means many. So it is not necessary that in all the cells, there is only one nucleus. The shape of the nucleus is normally oval, or it depends upon the type of the cell. It can also be cylindrical or horseshoe shaped, tri-lobed, branched, or irregular. The size of the nucleus varies with the cell type. It is directly proportional to the amount of cytoplasm that is found in the cell. The eukaryotic nucleus have a nuclear membrane, a nuclear pore, nucleoplasm, chromatin network, and nucleolus. We have already studied regarding this. So let us study about the nuclear membrane. It is an envelope that surrounds the nucleus. The outer membrane has got many ribosomes in it, and it is in continuous touch with the endoplasmic reticulum that is found in the cytoplasm. The nuclear membrane has got many pores, and these pores are not continuous. These pores help in entry and exit of materials inside the nucleus and outside the nucleus. The nuclear plasm is a semi-fluid that is found in the nucleus. It contains proteins, ions, enzymes, and nucleic acid. The chromatin network is found in the nucleoplasm of the nucleus. In the interphase nucleus, the chromosomes are fine threads and in the form of network. Interphase means when the cell is not dividing. So you can see over here how the chromatin looks like. This is a nuclear membrane. Then this is a nuclear pore. Pore means the holes. This is a chromatin. If you want to see how the chromatin looks like, see how the chromatin looks like. This is all it carries our, it is made up of DNA, and it carries our hereditary information from one generation to another. And you can see how the chromosomes looks like. NUCLEOLUS. It was discovered by Fontana. In almost all the cells in the interphase nucleus, nucleolus is present. It may be 1 or 2. In some cases, hundreds of nuclei will be present. For example, amphibian oocytes. And the size of the nucleolus is dependent upon the activity of the cell. They may be smaller or bigger. The nucleolus contains RNA, protein, enzymes. It is formed with the help of nucleolar organizing chromosomes. The function means it helps in the biogenesis of ribosome, and it helps in the process of mitosis. Function of the nucleus: it gives protection to the genome, helps in the reflection of DNA. It plays a major role in the general metabolism of the cell. It helps in synthesis of ribosomes, helps in synthesis of RNA, controls the synthesis of proteins. It is a seat of hereditary. This is a eukaryotic cell. Inside the eukaryotic cell, you can see the nucleus. Inside the nucleus, we have got a chromosomes. And these chromosomes are made up of the DNA. The full form of DNA is deoxyribose. The full form of DNA is deoxyribose nucleic acid. DE means no oxygen, just removed one oxygen. We have got RNA. The full form of RNA is ribose nucleic acid. So from ribose nucleic acid, oxygen is removed. Therefore, we call this a deoxy, DE-oxy, ribose nucleic acid. So deoxyribose. It is a double helical structure. See over here in the form of ladder. RNA has got only one, but DNA has got double helical structure. The prokaryotic cells are single cells, and they lack membrane-bound nucleus and other organelles. Prokaryotic cells are single cell. They lack membrane-bound nucleus and other organelles. It is derived from a Greek word, pro means before, and karyon means nucleus. I've been using this term again and again, and now you might have been used to the word prokaryotic. So pro means before and karyon means nucleus. They are considered to be the first living organism of the earth, and they are the simplest form of life. Examples are that of bacteria. The DNA which is carried by the prokaryotes is called as prokaryotic DNA. It is found in the cytoplasm of the bacteria. Prokaryotic DNA is found as circular plasmids. The prokaryotic DNA does not contain a nuclear membrane around it. It is packed into a single circular chromosome. It resides in the region called the nucleoid in the cytoplasm. These all are prokaryotic cells, and they all are bacteria. This is a bacteria. These bacteria are living in a colony. This bacteria is one only. They're living in a colony attached to each other, but it is a single cell organism. So the prokaryotic cell differs from the eukaryotic cells because they do not have any membrane. See how it looks like. So this is a DNA. It does not have any nuclear membrane around it. It has got a cell wall. The bacteria do have a cell wall. They have a flagellum. We have got only one flagellum that helps in movement of the organism. This mass of chromosome is called as nucleoid. You should know how to draw this prokaryotic cell. Appendages of prokaryote. The prokaryotic have got different types of appendages like the flagella we just saw, or a pili and fimbriae. The most common appendage, that is the organ that is used for movement, is called as flagella. It is a tail-like, and normally an organisms have got only one flagella. It is filamentous in nature. Pili, in turn, are longer filaments, and their roles are different. They help in the process of conjugation so that the DNA can be transferred from one of the organisms to another. This is a typical prokaryotic cell, and this is a typical eukaryotic cell. See, there's only one strand of DNA over here. The DNA are found inside the nucleus, and there are many strands, depends upon the organism. And there are organelles that have got membrane. Ribosome is found both in prokaryotic cell and also eukaryotic cells. Ribosomes do not have any membrane. Now, we have already studied about the nucleus and the plasma membrane. We are now going to study about cytoplasm. Where is the cytoplasm found? It is found between the nuclear membrane and the plasma membrane. It contains specialized type of cell organelles, and each of these cell organelles perform a special function for the cell. The organelle is a specialized subunit usually found within a cell, and it performs special functions. The name organelle comes from the idea that these structures are part of cells as organs are to the body. Let us study about different types of cell organelles. They are found both in plant cells and animal cells. This is a plant cell. Outside the plasma membrane is a protective structure that is called as a cell wall. The animals do not have any cell wall. You can see the vacuole is very large. The vacuole is very small. The vacuole is large and it has pushed the nucleus on the side. So there is one large vacuole. There are small vacuoles and they are more in numbers. Now we will be studying about different organelles in it. As we studied earlier also, the nuclear membrane is attached very much outside with the endoplasmic reticulum. So this is endoplasmic reticulum where it is attached with the nuclear membrane. So this is a nuclear membrane. See, with the pores, they have got a pores, and with the pores is attached in the endoplasmic reticulum. The endoplasmic reticulums are divided into two types. The rough endoplasmic reticulum, on which you can see these dots, they are ribosomes and organelle, and the smooth endoplasmic reticulum, which do not have any ribosomes on it. And the shape is also different for the smooth endoplasmic reticulum. These are smooth endoplasmic reticulum, and these are the rough endoplasmic reticulum. They function as a transport system of the cell. Let us go to the shape. This shape, that is flat shape, is called as cistern. Okay, these are vesicle shapes. Endoplasmic reticulum, short term we can also call it as ER. ER are of two types. They are called as rough endoplasmic reticulum and a smooth endoplasmic reticulum. Rough endoplasmic reticulum have ribosomes on it and they help in synthesizing protein. Smooth endoplasmic reticulums do not have any ribosomes. They help in synthesizing lipids. They are called as the highway of the cell. again as endoplasmic reticulum. This is a rough endoplasmic reticulum. These are smooth endoplasmic reticulum. How does transportation takes place in the cell with the help of endoplasmic reticulum? The ribosomes assemble proteins from polypeptides entering into the rough endoplasmic reticulum. So the ribosomes that are found over here, that organelle is made with the help of a nucleoplasm, and they are positioned on the rough endoplasmic reticulum. The materials that have been synthesized inside the nucleus can easily come out of the nucleus with the help of endoplasmic reticulum. It is a place where synthesis of proteins takes place and synthesis of lipid takes place and other steroids takes place in smooth endoplasmic reticulum. This shape is called as a cistern shape. You can see in the end a vesicle is formed. This is a vesicle also. Slowly and slowly, the product that has been formed by the synthesized by the endoplasmic reticulum is also there in the vesicle. It will be removed, it will detach itself from the endoplasmic reticulum and see a detachment. So then it will be transported to the Golgi body. That is another organelle. This shape is also a cistern shape. It will touch this. As soon as it touches this, the membrane will break open and the protein or the lipid or the steroids that it is transporting will enter inside the Golgi body. When it is entering inside the Golgi body, the storage work of these proteins and steroids or lipids is being done by the Golgi body. So synthesizing of proteins, lipids is being done by the endoplasmic reticulum, the proteins by the rough endoplasmic reticulum, the lipids by the smooth endoplasmic reticulum. The storage is being done in the Golgi complex. We also call it as Golgi apparatus. The Golgi apparatus, similarly, what they will do? They will store and they can send in the same form, or else they can even modify those proteins, and according to the need inside the cell, they will again send it to their vesicle. It will break and they can send to any part of the organelle or even outside the cell. So this is how transportation in cell takes place. The vesicles travels to the cell membrane, fuse with the cell membrane and release the protein to the outside if it is to be sent outside. If it is to be sent to other organelle, it will move to the other organelle. To the mitochondria, yes, it will move to the mitochondria, and when it travels to the mitochondria, it fuses with the cell membrane of the mitochondria and it will release the protein on it. But mitochondria has got its own DNA and it synthesizes its own protein also. So transportation in cells, how did it take place? See, the rough ER, the vesicle is formed, the vesicle from the ER, it comes to the Golgi apparatus. From the Golgi apparatus, it is a storage house. A vesicle from the Golgi body can move to the outer part of the cell membrane, or it can also move to the vacuole. It can also move to the lysosomes or other part of the cells. So this is how transportation takes place in the cell. The function of rough endoplasmic reticulum, what is the function? It modifies protein and this protein will be shipped to other locations inside the cell or outside the cell. Rough endoplasmic reticulum, the shape is cistern and it is called rough because it is called ribosomes on it. This is a smooth endoplasmic reticulum. No ribosomes is found on it. It synthesizes fatty acids, phospholipids, steroids. It deoxyphizes. काट देना. Golgi apparatus. It is called as a post office. Likened to the cell's post office, the Golgi apparatus modifies, sorts, and packages protein for secretion. So whichever protein has to be sent in packages, it will send only that protein to that particular part. Therefore, it will sort out and then it will pack in the vesicles and then send it. Okay. The Golgi apparatus or the Golgi body or the Golgi complex or simply Golgi. You can give it any name to it. It is a cellular organelle present in most of the cells of the eukaryotic organisms. It is made up of flattened stacks that are called as cisterns. It is involved in packaging of protein molecules before they are sent to their destination. These organelles help in processing and packaging the macromolecules like proteins and lipids that are synthesized by the cell, and therefore they act as the post office of the cell. The Golgi apparatus was discovered by Camillo Golgi. Structure of Golgi apparatus. Under the electron microscope, the Golgi apparatus is seen to be composed of stacks of flattened structures that under the electron microscope, the Golgi apparatus is seen to be composed of stacks of flattened structures that contain numerous vesicles containing secretory granules. The Golgi apparatus is a complex array of interconnecting tubules, vesicles, and cisterns. Interconnecting tubules means here. These all are these are interconnected, no? So here also, like in endoplasmic, interconnection is there. Here we cannot see, but yes, there is interconnection over here. All the cisterns are interconnected to each other. It is the simplest unit of Golgi apparatus. Golgi apparatus is seen as a stacks of flattened structures, and these flattened structures are called as cisterns. All these cisterns are interconnected to each other with the help of a tubule. They got a vesicles also along with them. I'll show you all these things: cistern, vesicles, and tubules. Here you can see this is flattened structure, and these flattened structures are called as cisterns. These round structures that are found, they are called the vesicle parts. This vesicle and these cisterns are attached to each other with the help of tubules, and even these flattened structures are connected to each other with the help of tubules. You can see here in the endoplasmic reticulum, this is a tubule, this is a cistern, and this is a vesicle. So you should know the shape. This is a vesicle, again I'll repeat, this is a cistern, this is a tubule. So all, even in Golgi body, all these flattened cisterns are interconnected to each other with the help of tubules. Even you have got tubules between the vesicle and the cistern. Golgi body is involved in packaging of the proteins, and each of this cistern is flattened plate-like, and they are held in parallel bundles or stacks, one above the other. In Golgi apparatus, such stacks are nearly 5 or 6 in numbers. Lysosome. Lysosome is found in cytoplasm. It is filled with digestive enzymes. The function is, this digestive enzyme helps to break down the complex food into simple food. The other function is, it removes the junk from the cell. See, it is removing the waste from the cell. Therefore, it is also known as cleanup crew. But sometimes a human can have diseases. The lysosomes fail. The normal function cannot be performed. And what happens? The lysosome spreads out and it will start digesting all the healthy organelles also. And therefore, the cell dies. So this is how you can see in a lysosome. This is a this is a lysosome. Inside this, there is a digestive enzyme. If you have to draw as a figure, you can draw like this. Then, as a foreign particle is entering inside, it is being attracted towards the lysosome. It will enter into the lysosome. See, it is merging. And what happens? It will remove the waste over here. And whatever materials that it have changed, that is from a complex to simple, it can be used in the form of energy. Lysosome, you have to draw like this. It has got a membrane and it has got enzymes. Those enzymes are called as digestive enzymes. Lysosome, see, a bacteria is entering inside. We got a receptors that senses it. It will enter inside a vacuole like this. It enters in the lysosome and the bacterium vacuoles, they merged. The digestive enzymes will try to digest it, and whatever the waste is, it will throw out. It is called as exocytosis. It is also, maybe this is a food for an organism, then it is called as phagocytosis. Phagocytosis means taking inside the materials, and this is taken inside it. And what happens? Here it is thrown out. Now, how is it thrown out? As soon as this membrane touches this membrane, it will break open it, and the waste is thrown out. Lysosome can work as a suicidal bag. If the cell gets damaged, then one of the lysosome bursts and it will release the digestive enzyme. These released enzymes will digest their own cell, and untimely the cell dies. And therefore, lysosome is also called as a suicidal bag of the cell. You can see over here, lysosome, how it is engulfing the mitochondria. It has killed the mitochondria. It has digested the cell. Let us study about another organelle, that is mitochondria. We have learned that it's double membrane. See the outer membrane, it is flat. And see the inner membrane, it is highly folded. This highly folded part is called as cristae. The matrix is found inside this double membrane layer. It has got its own ribosomes and a DNA. It can synthesize its own protein. It will not require a ribosome to come in the protein synthesis that it has done. It can synthesize its own DNA because this is a mitochondria. This is an outer membrane that is flat. This is an inner membrane that is highly folded. These folds are called as cristae. Inside the cristae, you can see the materials that are found inside the mitochondria. They are the ribosomes. They also have their own DNA. Therefore, they have their own genetic material. They can synthesize their own protein. The mitochondria has got an inner and outer membrane. The two membranes have different properties. The outer membrane is continuous. The inner membrane is highly folded. Highly folded means to increase the surface area. And that inner folding, because it has increased the surface area, it is the power called as the powerhouse of the cell. The surface area to perform various activities to release energy can easily be done. Therefore, the mitochondria is called as the power of the cell because it produces the energy currency of the cell, ATP, through the process of respiration. Another double membrane organelle is a plastid, and it is found only in a plant cell. The functions are storage of food, synthesis of materials. Plastids are of different types. If a green pigment is found, that is a chlorophyll is found, it is called as a chloroplast. If a plastid contains pigments apart from green, it is called as a chromoplast. You can see different flowers of different colors. There, it is not that a plant is always green in color, it has got flowers of different colors because they are rich in chromoplast. And if a plastid does not have any pigments, it is white in color, colorless, it is called as a leucoplast, and it is mainly involved in the storage of the food. Chloroplast, the shape is like this. Chromoplast, it looks like this, and leucoplast. It contains chlorophyll, produces and stores glucose. It contains carotenoids, that is a red, orange, and yellow pigments. It is found in the flowers and the fruits. It contains no pigment and stores starch. You should know the structure of the chloroplast. A chloroplast is made up of two parts mainly. The stroma part, this is the stroma part, and these convex structures are called as the grana. So a chloroplast has two parts, a grana, which is found in the stroma. A grana looks like a pile of coins. The unit of grana is thylakoid. This all is called grana, it is plural. But if I talk about one, I have to say granum. So I'll repeat again. This is a granum. The unit of this granum is thylakoid. This granum, this is a granum, this is a granum, and this is granum. All together are called as grana. It is a double membrane. It has got its own DNA, like which said about mitochondria. So in exams, a question can be asked, which organelles have their own DNA, a genetic material? There are two. You can say a chloroplast, a plastid, and a mitochondria. Each granum has number of disc-shaped sacs called as grana lamellae, or you can also call as thylakoids. These thylakoids are piled one over the other. All these grana are interconnected with the help of intergrana. I'll show you the intergrana. This is intergrana. They are interconnected with each other. Single thylakoids called stroma thylakoids are found in the chloroplast. The last organelle that I will be covering today is vacuole. It is present in all plants, fungal cells, protists, animals, bacterial cells. They are filled with water and also contains inorganic and organic molecules. They also include enzymes that are found in the solution. In certain cases, they may contain solids also, which have been engulfed. Vacuoles are formed by the fusion of multiple membrane vesicles and are effectively just larger form of these vesicles. The organelles have no basic shape or structure. Its structure varies according to the requirement of the cell. This is a vacuole. The membrane of vacuole is also called as tonoplast. It is small and round in animals and large single bag that in plants. In plants, when the cell was very young, there were many vacuoles. They all merged, and when the cell is matured, they've got a single large vacuole in it. And this vacuole pushes the nucleus towards the side. Most mature plants have large vacuole, and it occupies 30% of the cell's volume and can occupy as much as 80% of the volume, certain cell types and conditions. Strands of cytoplasm often run through the vacuoles. A vacuole is surrounded by a membrane called tonoplast, and it is filled with cell sap. Animal vacuoles are smaller than their plant counterparts, and they're greater in number. There are also animal cells that do not have any vacuoles. So vacuoles of animal cells and plant cells. Several small vacuoles. They are used for storage and they contain nutrients, water, or the waste. As I told you, they can have nutrients because they have got they can store the nutrients that is found in the cell. They also contain waste over here, which can be thrown out by exocytosis, or they can have water to maintain the water balance of the cell. The plant cells have got one large vacuole, and it is used to store water and push against the cell wall. It keeps the plant rigid. A sac of fluid surrounded by a membrane is the structure of a vacuole. It is used for temporary storage of waste, nutrients, and water. The plant cells and animal cells have many organelles in common. Which are the common organelles that you can see over here? A nucleus, then a nucleoplasm it has, endoplasmic reticulum, both are having, then mitochondria, both are having, then it have got centrioles. No, it do not have centrioles. Centrioles have cell divisions. These are centrioles. They have cell divisions. They have got ribosomes. Even it has ribosomes. Lysosomes. Yes, lysosomes are also present over here. And it has a cell membrane. It also has a cell membrane. It has got a cell wall. And it does not have a cell wall. Animal cells do not have a cell wall. So plant cells have got cell wall. They've got a plastids, chloroplast. Animal cells do not have cell wall or plastids in it. Animal cells have centrioles, plant cells do not have centrioles. And other differences regarding the vacuole. The vacuole cell is so big and vacuoles are small and few in number. So these are the differences and similarities between the plant cells and animal cells. They have nucleus, cytoplasm, plasma membrane, Golgi apparatus, mitochondria, endoplasmic reticulum, and ribosomes. Animal cells do not have fixed shape. They have a fixed shape. They do not have cell wall. They have a cell wall. They do not have vacuoles. If they have, the vacuoles are very small. They have large vacuoles. They do not have chloroplast. They have chloroplast. The food is stored in the form of glycogen. The food is stored in the form of starch. They have centrioles. They do not have centrioles. So you all should know the difference between the prokaryotic and eukaryotic organisms. The prokaryotic cells look like this. It has a nucleus. It has a well-developed nucleus with a nuclear membrane. So you can talk the difference between the prokaryotic and the eukaryotic on the basis of the membranes, on the basis of the size of the cell, nucleus, on the basis of membranes, bond organelles, flagella, then glycocalyx on the basis of cell wall, plasma membrane, cytoplasm, and ribosomes, chromosomes, cell division, and how sexual reproduction takes place. So these are the points that will help you to write the difference between the prokaryotic organisms and eukaryotic organisms. We have studied almost most of the differences. Cell division is a process by which a parent cell divides into two or more daughter cells. The main types of cell divisions are mitosis and meiosis. This is mitosis. Let us take that in a mitosis, the number of chromosomes are 46. After cell division, it will give rise to two daughter cells, and they are diploid natured. This is diploid means 23 pairs. So here the diploid status remains. The two daughter cells have got same number of chromosomes as in the parent cell. In meiosis, the process is long. The cell was diploid. It has got 46 chromosomes, that is 23 pairs of chromosomes. During the first cell division, the number of chromosomes remains the same, that is, it remains diploid. But during the second cell division, the number of chromosomes becomes half. And in the end of the meiosis, we have got 4 daughter cells, and they are haploid in nature. The number of chromosomes are half. So in mitosis, the daughter cells are diploid in nature. In meiosis, the daughter cells are haploid in nature. The number of 2 daughter cells are formed. The number of 4 daughter cells are formed. It is a simple one-step process, not a one-step process. But it is a simple process, which is a very long and complex process. This mitosis takes place in somatic cells of our body or in any living organisms. This meiosis takes place in the any types of only the gamete cells of the organisms. So mitosis takes place in somatic cells. Meiosis takes place in the gamete, in the formation of, helps in formation of the gametes. Mitosis produces genetically identical cells. They result in diploid cells. They take place throughout an organism cells, and they are involved in asexual reproduction. Meiosis, they produce genetically unique cells. They result in haploid cells. They take place only at certain times in an organism's life cycle, and they are involved in sexual reproduction. Thank you so much.